Usually, when we picture the historical record, right, we imagine it as this entirely static thing. Like, we envision events carved permanently into, I don't know, limestone monuments or
Hmm.
written on fragile parchment that's disintegrating, or maybe buried under layers of volcanic ash.
Right. Waiting for an archaeologist with a trowel to find it.
Exactly. With a tiny brush, just slowly scraping away the centuries. We tend to view history as this external artifact, something outside of ourselves.
It's a really pervasive way of thinking, actually. We naturally externalize the past. We treat it as a series of events that happened to, other people in other places a really long time ago. We totally separate the historical narrative from our present physical reality.
Which, makes sense until you look closer, because some of the most meticulously detailed, brilliantly complex, and entirely unedited history books in existence are not sitting on a climate-controlled shelf in some museum archive.
No, they're not.
They are actively pulsing through your veins right now. Every single cell in your body contains this unbroken logbook of ancestral journeys, surviving famines, ancient migrations, and thousands of years of human interaction.
And shifting your perspective to view human DNA as a historical document, well, it changes everything. It's a molecular ledger.
A molecular ledger. I love that.
Yeah, because when we sequence a genome, we aren't just looking at biological instructions for eye color or protein synthesis. We are literally reading the surviving data of our ancestors' geographic and social movements. We're tracking who they met, where they migrated, and who they formed communities with.
And that biological ledger is precisely what we are cracking open today. So welcome to the Deep Dive. I'm your host, and today we are shrinking down to the microscopic level to resolve some incredibly massive, centuries-old historical debates.
And I'm thrilled to be here to unpack this with you. The roadmap for our exploration today is a brilliant piece of research. It's titled Molecular Insight into the Genesis of Ranked Caste Populations of Western India.
Right, published by researchers Sonali Gaikwad and V.K. Kashyap. And let me tell you, reading through their findings, it feels less like reading a standard genetics paper and more like, I don't know, watching a high-stakes, multi-millennial detective story unfold.
The methodology and the ambition of Gaikwad and Kashyap's work are truly remarkable. Our mission for this deep dive is basically to follow their lead, right? We're going to utilize this extremely high-resolution genetic data to essentially fact-check the historical, cultural, and mythological origin stories of 4 distinct social groups in Western India.
Fact-checking ancient myths with DNA. It's wild.
It really is. The researchers sought to understand how these populations moved, how they segregated themselves, and, importantly, how societal rules of marriage and class interacted with the fundamental biological imperative to mix.
So for you listening, if you find yourself fascinated by history, by the mechanics of genetics, or simply by the profound complexities of human nature, this deep dive is going to fundamentally rewire how you perceive human boundaries.
Oh, absolutely.
We're going to examine social walls that have been described historically as these rigid, impenetrable fortresses, and we're going to see through the lens of molecular biology just how permeable those walls actually are.
We're going to prove that human biology possesses a momentum entirely its own.
But to fully appreciate the twist ending of this genetic analysis, and there's a massive twist, we first need to establish the socio-historical canvas we're analyzing. We have to understand the specific, highly stratified world these genes exist within.
Right, because context is everything here.
Yeah.
You simply cannot interpret the genetic landscape of the Indian subcontinent without first grappling with its staggering, almost unfathomable demographic scale.
We are looking at a state of megadiversity.
Total megadiversity. For tens of thousands of years, the subcontinent has functioned as this massive geographic funnel, a melting pot for incredibly varied genetic pools. You have the ancient original inhabitants populations, often categorized anthropologically as proto-Australoid groups.
Okay, so they were there first.
Yes. Layered upon that, you have Caucasian Mediterranean influences arriving from the west. Then you have Mongolian genetic strains filtering in through the northern and eastern mountain passes. It's a composite formed from dozens of distinct ethnic strains arriving and interacting over millennia.
And while that slow, continuous blending was happening for, tens of thousands of years, the paper points to a specific, highly disruptive demographic event that drastically altered the social architecture of the region.
Yes, a massive shift.
We're talking about the Holocene period, right? Roughly between 4000 and 1500
Yeah.
BC.
Exactly. That time frame marks a monumental demographic and cultural shift. This is the era associated with the arrival of the Indo-Aryan tribes.
Right.
These were pastoral, nomadic groups speaking Indo-European languages, and they were migrating downward from the steppes of Central Asia. They carried with them a specific cultural and technological package that archaeologists identify as the Andro Novo culture.
And the Andro Novo culture wasn't just like a group of wandering shepherds, though. From what I understand of the archaeological consensus, they were riding in with some massive technological upgrades.
Huge upgrade.
They had mastered horse-drawn chariots, right, and they possessed superior metallurgy, particularly when it came to weaponry.
Yeah, their military and technological advantages were highly significant. And when they migrated into the Indian subcontinent, they initiated this massive wave of cultural diffusion.
How massive are we talking?
To give you a sense of the scale of their impact, Indo-European languages eventually spread to encompass roughly 3/4 of the contemporary Indian population.
Wow, 3/4. That's a profound impact.
It is. However, this arrival didn't just change how people spoke. It really catalyzed a deep structural stratification of the society. Broadly speaking, the population fractured into two parallel tracks.
Okay, what were the tracks?
Well, first you had the autochthonous tribes. These were the indigenous groups who essentially retreated to forested or mountainous regions. They maintained their traditional, localized ways of life. And today, these indigenous tribal groups make up about 8% of the population.
And the other track?
The rest of the population began to organize into what eventually became the Hindu caste fold.
And that caste fold, which eventually encompassed roughly 80% of the population, is the primary target of our deep dive today. Now, the classical texts define this system in 4 broad hierarchical layers, which were initially tied to occupation, right?
Correct. Broadly based on occupation.
So at the apex, the top of the pyramid, you have the Brahmin, the priestly, scholarly, and administrative class.
Right.
Below them are the Kshatriya, the aristocratic and warrior class. Then you have the Vaishya, encompassing the landowners, merchants, and traders. And finally, at the foundational layer, you have the Shudra, the agriculturalists, laborers, and service providers.
That's the classical framework. But the crucial mechanism to understand here isn't just the division of labor. It's how that division was locked into place.
Right, because it didn't stay just about your job.
Exactly. Over centuries, what began as occupational categories solidified through the strict enforcement of endogamy.
And for those who might not be familiar, endogamy is basically the practice of restricting marriage exclusively to individuals within your own specific local group or caste, right?
Yes, exactly. By mandating these marriage rules generation after generation and enforcing them through religious texts and immense social pressure, the caste system theoretically created an incredibly complex genetic substructuring.
Like trying to build walls inside the gene pool.
That was the goal. To build hundreds of separate, isolated genetic islands that were all sharing the exact same geographic landmass.
It's wild to think about. Like, if we think of the caste system as a vast, ancient metallurgical experiment, the architects were trying to create distinct, unmixed alloys. If you were born a Brahmin, your biological destiny was sealed. You would only ever mix your genetic material with another Brahmin.
That's the ideological theory, yes.
But biology naturally resists that kind of isolation, doesn't it? I mean, even when you look at the historical texts, especially concerning the middle ranks like the Kshatriya warrior class, you can see the system having to bend to accommodate reality.
Oh, the warrior class is a perfect example of this systemic adaptation. The researchers highlight the Kshatriya because their rank was, out of necessity, highly adaptable.
Why out of necessity?
Well, think about the geography. As wave after wave of incoming Caucasoid invaders pushed into the subcontinent over the centuries, groups like the Greeks under Alexander, the Parthians, the Scythians from the Eurasian steppes, the Kushans, they brought formidable military power. The existing social structure had to figure out what to do with these powerful, armed foreign populations who were deciding to stay. You couldn't just categorize heavily armed conquerors as laborers or priests.
Exactly. If a massive, heavily armed cavalry rolls into your territory, conquers the local leadership, and just establishes themselves as the new rulers, the existing societal framework has to adjust. You can't just hand them a broom. You essentially have to assimilate them into the warrior cluster.
Right. Precisely. They were socially absorbed into the Kshatriya fold. Consequently, from a theoretical standpoint, the warrior class should represent a highly mixed, fluid genetic group compared to the more insulated priestly or laboring classes. It was a pragmatic social absorption that inevitably led to genetic absorption.
Which makes total sense.
Mm.
Now, this brings us to a really important geographical question. The Indian subcontinent is vast. I mean, it has massive river valleys in the north, dense southern jungles, enormous deserts. Why did Gaikwad and Kashyap choose to focus their genetic lens specifically on Western India?
It's a very deliberate choice.
Right, they zeroed in on the state of Maharashtra, specifically the Konkan coast and the North Deccan Plateau. What makes this specific terrain the ideal laboratory for testing these genetic boundaries?
Western India presents a really unique confluence of geography and history. It serves as the perfect natural laboratory because, quite simply, it was never isolated.
Ah, okay.
Historically, the Deccan Plateau was the crucial terrestrial junction. The major caravan trade routes connecting the northern empires to the southern kingdoms all had to pass through this region.
Let's go crossroads.
Exactly. But perhaps even more significantly, the Konkan coast was a massive ancient hub for international maritime trade. From roughly 1500 BC stretching all the way to 1200 AD, the ports along this coast were actively engaged in commerce with highly advanced Western Eurasian cultures.
We're talking about direct, sustained maritime contact with Greece, the Roman Empire, and the Iranian Plateau, right?
Yes, deep and sustained contact.
I actually recall reading about the monsoon wines, the Hippolytus wines, I think the Romans called them, which allowed merchants to sail directly across the Arabian Sea, like from the Red Sea straight into the Konkan coast.
Yeah.
They weren't just hugging the shoreline, it was an oceanic highway.
An oceanic highway is a great way to put it.
And they were creating black pepper, textiles, and spices for Roman gold and wine. And, with that level of massive maritime infrastructure, you aren't just exchanging cargo, you're exchanging human beings.
That is the exact dynamic that makes this region so vital to the study. You had sailors, merchants, envoys, and sometimes entire communities of expatriates setting up long-term residences in these coastal port cities.
Wow.
This rich, thoroughly documented history of Western Eurasian immigration sets a really fascinating stage. You have this deeply stratified local caste system enforcing strict rules of endogamy, sitting directly on top of one of the busiest, most cosmopolitan ancient international trade routes in the world.
So the researchers wanted to answer a fundamental question. When you clash the strict, isolating rules of the caste system against the constant, chaotic influx of foreign travelers and merchants over thousands of years, what actually happens at the molecular level?
It is the ultimate test of ideology versus biology.
So how do you actually measure that?
Yeah.
How do you solve a demographic mystery that spans millennia? You obviously need a highly specific set of tools. Gaikwad and Kashyap didn't just, take a generic DNA swab and look for broad similarities.
Oh, no, not at all.
They utilized a deeply rigorous methodology, employing what is essentially a three-dimensional molecular toolkit. They examined 3 entirely distinct types of genetic markers to build their historical model.
Right, because to accurately reconstruct a population's ancestry, a single genetic marker is wildly insufficient. It only gives you one flat angle of a very complex, multidimensional object. The researchers here utilized 3 different genetic systems, each operating under different biological rules, to ensure they were capturing the complete historical narrative.
Let's clearly define these 3 lenses, because understanding the mechanics of how we track this DNA is crucial to making sense of the wild conclusions the researchers eventually reach. The first lens they used focuses exclusively on the paternal Y chromosome.
Yes, lens 1. Now, the Y chromosome is unique in our genome because it is passed down almost exclusively from father to son. In the realm of genetic anthropology, it's considered non-recombining.
Meaning it doesn't mix.
Exactly. Unlike the rest of your DNA, which is a jumbled mix of your mother and father, the majority of the Y chromosome does not cross over or mix during reproduction. It is handed down intact generation after generation. It serves as an unbroken chain back through the male lineage. The only changes that occur are occasional random mutations.
And the researchers targeted 2 specific types of mutations on the Y chromosome, right? YSNPs and YSTRs. To understand how they reconstruct timelines, we need to look at how these 2 mutations function differently.
That distinction is paramount. Let's start with SNPs, which stands for single nucleotide polymorphisms. You can think of an SNP as a very rare, very slow mutation. It's like a single spelling error in the DNA sequence that happens maybe once every few thousand years.
Got it.
Because they are so rare and so stable, SNPs act like the hour hand on a molecular clock. By analyzing 20 specific YSNPs, the researchers could identify broad, ancient paternal lineages called haplogroups. This allows them to trace a male line back tens of thousands of years to massive continental migrations.
Okay, so SNPs give us the ancient macro-level picture. They tell us if your paternal ancestor was a hunter-gatherer in Europe or an early farmer in the Levant. But then we have STRs, right? Short tandem repeats.
Yes, STRs are the minute hand or even the second hand of the molecular clock. An STR is a short sequence of DNA that repeats itself, almost like a molecular stutter. And because the cellular machinery sometimes slips when copying these repetitive sections, STRs mutate much faster than SNPs. They might gain or lose a repeating unit every few generations. Right, so by analyzing 20 different YSTR loci, researchers can zoom in past the ancient, broad haplogroups and identify specific, much more recent familial relationships. It provides a high-resolution snapshot of recent paternal genetic drift.
So together, SNPs and STRs give us the complete, unbroken, deep-time history of the male line.
Exactly.
That covers the paternal ancestry, lens 1. Lens 2 looks in the exact opposite direction. It traces the unbroken maternal line using mitochondrial DNA, or mtDNA.
Right. While the Y chromosome lives in the nucleus of the cell, mitochondrial DNA exists in the mitochondria, which are the energy-producing structures outside the nucleus. And crucially, you only inherit your mitochondria from your mother's egg cell, never from the father's sperm.
So it's the exact mirror of the Y chromosome.
Exactly like the Y chromosome, mtDNA is non-recombining. It is passed exclusively from mother to child. The researchers examined 18 highly specific diagnostic sites and the hypervariable regions HVS1 and HVS2, which mutate at a known rate. This allowed them to trace the unbroken female line back to the ancient maternal founders of these populations.
Now, before we move to the third lens, there is a sociocultural phenomenon discussed in the paper that fundamentally alters how we have to interpret this male and female DNA data. The researchers heavily emphasize the concept of patriarchality.
Yes. Very important.
When I read that section, it completely clarified why geneticists often find such wildly divergent stories between paternal and maternal lines in the exact same population. Can you unpack patriarchality for us?
Absolutely. Patriarchality is a foundational concept in human anthropology. In many traditional societies across the globe, and particularly within the historical structures of the Indian subcontinent, marriage customs dictate that the woman leaves her family home to reside with her husband's family and his broader community.
So the men stay put.
Yes, the men are geographically stationary, they inherit the land, they stay in their ancestral villages, and they build their social power locally. The women are the ones who migrate.
And if you extrapolate that social custom over 4,000 or 5,000 years, it creates a massive structural bias in the genetic record, doesn't it?
It creates a tremendous asymmetry because women are consistently moving between villages, between regions, and occasionally across broader social boundaries upon marriage. Maternal DNA, the mtDNA, tends to get shuffled around and shared across different populations much more fluidly.
It acts like a genetic bridge.
Exactly, a genetic bridge. Meanwhile, the paternal Y DNA stays geographically locked. The men act as stationary genetic anchors. This is why, when researchers look at these populations, they frequently find that the differences between groups are far more pronounced in their Y chromosomes than in their mtDNA.
Because the paternal lines reflect the strict geographic and social borders, while the maternal lines reflect the continuous, quiet flow of human migration.
Precisely.
That perfectly sets up Lens 3. We have the stationary fathers and the migrating mothers. But Lens 3 isn't looking at unbroken lines at all. It's looking at the great genetic mashup. This is autosomal DNA.
Yes. The researchers utilized 15 highly polymorphic biparental autosomal STRs. Autosomal DNA makes up the vast majority of your genome. It is the DNA found on your 22 pairs of non-sex chromosomes.
And this one mixes.
Right. Unlike the Y chromosome or mtDNA, autosomal DNA actively recombines. During meiosis, which is the process that creates sperm and egg cells, the chromosomes you inherited from your mother and father cross over and swap segments. You inherit exactly half from your mother and half from your father, but it's a random shuffle of their combined ancestries.
So unlike the first two lenses, which are tracing a single thread through a massive tapestry, autosomal DNA is looking at the entire fabric of the tapestry as it exists right now. It doesn't give us a clean, unbroken, ancient line. Instead, it provides an incredibly sensitive, highly accurate picture of the overall recent mixing and the total genetic contribution from all sides of a person's family tree.
It measures the current biological reality.
Okay, let's synthesize the toolkit so everyone is on the same page. Lens 1, the Y-DNA, acts like a paternal surname, right? It's passed down unaltered straight from father to son for centuries, carrying the legacy of a specific ancient patriarch.
Perfect analogy.
Lens 2, the mtDNA, is like a maternal family heirloom.
Yeah.
Perhaps a specific piece of jewelry, handed directly from mother to daughter for thousands of years.
Yes.
And Lens 3, the autosomal DNA, is the family recipe book. It contains a pinch of spice from the father's side, a specific technique from the mother's side, and it represents a total pragmatic blend of everyone who has contributed to the family's history over the last several generations.
That's exactly how it works.
So armed with that three-dimensional toolkit, the surname, the heirloom, and the recipe book, Gaikwad and Kashyap proceeded to sequence the DNA of 365 individuals, drawn from 4 highly specific, historically entrenched castes in Maharashtra.
Right. And they then compared those genetic profiles against large databases of Central Asian, West Asian, and European populations.
And the moment they applied this toolkit to the absolute pinnacle of the social hierarchy, they uncovered a massive historical contradiction. Let's look at the Brahmins.
The study focused on 2 distinct Brahmin subgroups. You have the Dasas Brahmin and the Chitpavan Brahmin. Now, within the theoretical framework of the caste system, both of these groups occupy the absolute highest tier.
The very top.
The very top. They are the religious elites, they are the custodians of sacred knowledge, and they comprise a very small fraction of the overall population in Western India. They share the same elevated rank.
However, despite sharing the same VIP status, anyone familiar with the regional history knows that culturally, linguistically, and historically, the Dasas and the Chitpavan are distinctly different populations.
They are profoundly different. The Dasas Brahmin represent the older, deeply entrenched establishment. They are Marathi-speaking, their history is woven into the very fabric of the region, and they possess an incredibly complex, extensive internal system of subdivisions known as gotras.
A gotra being like a clan system.
Essentially, yes. It's an exogamous clan system used to prevent inbreeding within a caste. And having such a mature, elaborate gotra system strongly indicates that the Dasas have been present in the region, consolidating their social position and managing their genetics for a very, very long time.
Okay, so that's the deep ancient establishment. Now contrast that with the Chitpavan Brahmin.
The Chitpavan Brahmin presents a complete historical enigma. First, they speak Konkani, which is a distinct regional language. Geographically, they are tightly clustered around the relatively isolated Konkan coastal strip.
Okay.
But the real mystery is their ethnohistory. If you attempt to trace the Chitpavan lineage back through the historical records, their trail effectively vanishes into thin air around 1,000 years ago.
They just appear.
They just appear. Furthermore, physical anthropologists have long noted phenotypic distinctions. Historically, the Chitpavan population has frequently exhibited features more commonly associated with Western Eurasian populations.
Like what kind of features?
A more Nordic skeletal build, for one, and a notably higher incidence of lighter-colored eyes, which stands in stark contrast to the surrounding indigenous demographic.
So we have 2 groups, sitting side by side at the very top of the social pyramid, but presenting 2 entirely different cultural and physical profiles. Applying the genetic toolkit to the Dasas Brahmins first. Given their deep establishment in the region, I would expect their DNA to show a classic Indo-Aryan genetic signature, right? Like a straight, unbroken line back to the Central Asian steppes.
Well, if you look exclusively at their paternal lines, your expectation is perfectly met.
Oh, really?
Yes. The Y-DNA of the Dasas Brahmin revealed a remarkably high frequency of a haplogroup designated as R1A1. In the world of population genetics, R1A1 is the quintessential signature of the Indo-European expansions. It has a profound affinity with Central Asia and the Pontic-Caspian steppe.
Okay, so the paternal line tracks.
Exactly. The data unequivocally supports the historical narrative that the paternal ancestors of the Dasas were part of those early chariot-driving Indo-Aryan migrations into the subcontinent during the Holocene.
So the fathers, the possessors of the surnames, came over the mountains from Central Asia. But what happened when the researchers checked the maternal heirlooms? Did the women come from the steppes as well?
This is where the simple narrative fractures.
Uh-oh.
Their maternal mtDNA does not mirror the paternal origins at all. Instead of Central Asian lineages, the Dasas women predominantly possess Paleolithic M haplogroups.
Wait, Paleolithic.
Yes. To put that in perspective, using the molecular clock we discussed earlier, the M haplogroup represents some of the most ancient, deeply rooted, indigenous South Asian maternal lineages. We're talking about genetic signatures that have been present on the subcontinent since the earliest waves of human migration out of Africa.
Wait. If the men are migrating from Central Asia, but the women carry DNA that has been in India since the Paleolithic era, how does a population with that specific genetic division end up forming the elite priestly caste?
It reveals a fascinating mechanism of ancient social engineering. Anthropologists refer to this as the upward social mobility of indigenous women in deep antiquity.
Okay, break that down for me.
When those early Indo-European tribes migrated southward from the harsh Central Asian steppes, it was likely a highly male-biased migration.
Mostly warriors and herdsmen.
Exactly. They simply did not bring enough women with them to sustain a growing population in their new territory. Consequently, these incoming Central Asian men who were utilizing their technological superiority to establish themselves at the apex of the newly forming social hierarchy married local indigenous South Asian women.
Wow. And over thousands of years, that pragmatism forged a completely composite population.
No.
The researchers actually explicitly coined a term for this genesis, don't they? They call it Scytho-Dravidian.
Yes, Scytho-Dravidian. It's a literal biological synthesis of incoming Scythic or Central Asian male lineages with ancient Dravidian or indigenous South Asian female lineages.
So the Desath are living proof that this supposedly pure elite caste was actually a profound genetic hybrid from its very inception.
Yes, that Scytho-Dravidian model perfectly explains the Desast. But when the researchers turned their instruments toward the mysterious Chitpavan Brahmins, the ones with the missing history and the light eyes, they discovered a genetic profile that entirely defied the ancient Central Asian model.
Let's look at their paternal surname first.
Okay.
Did they carry the R1A1 marker of the ancient conquerors?
Not quite. They showed a high frequency of a related but distinct lineage, R1A, but critically, they also possessed a significant frequency of haplogroup L.
Haplogroup L.
Yes, and when the researchers examined their maternal mtDNA, the divergence became massive. The Chiptavan did not possess the ancient Paleolithic South Asian M lineages that anchored the Desas to the deep history of the subcontinent.
No ancient Indian maternal roots.
No. Instead, their maternal genetic pool was dominated by much younger haplogroups, specifically HV and U3.
And tracing those HV and U3 maternal markers on the global genetic map, where do they originate?
They are unmistakably Mediterranean and Eastern European maternal lineages.
Mediterranean. That's incredible.
It is. The researchers took all this multi-locus data and ran it through a principal component analysis.
TCA.
Right, PCA. It's a powerful statistical technique that reduces massive amounts of complex data into a visual graph. It plots populations based on their total genetic variance, so populations that share deep ancestry clustered together on the graph, while genetically distinct groups are pushed far apart.
Makes sense.
On this PCA plot, the Chitpavan Brahmins appeared as a stark outlier. They did not cluster tightly with the other Indian castes. Instead, they showed striking genetic affinities with populations thousands of miles away.
Which populations?
Specifically Iranians, Ashkenazi Jewish populations from Turkey, and Greeks.
Wow. If the PCA is clustering them with Greeks and Anatolians, that strongly suggests they didn't walk over the Hindu Kush Mountains thousands of years ago with the Indo-Aryans. They had to have arrived via a completely different vector.
All genomic evidence points away from the ancient terrestrial migrations and toward a much more recent maritime arrival.
The ports.
Exactly. The data beautifully correlates with the theory that the ancestors of the Chitpavan Brahmin originated in Anatolia, modern-day Turkey, or the broader Eastern Mediterranean. Given their sudden appearance in the historical record roughly a millennium ago on the geographically isolated Konkan coast, it is highly probable they migrated via sea vessels.
So they were sailors.
They were likely sailors, merchants, or perhaps a community fleeing religious or political persecution in the West, seeking refuge and new economic opportunities along the established Indian Ocean trade routes.
But arriving on a beach as a foreign merchant is one thing. How do you go from being an Anatolian sailor to occupying the highest echelon of the Hindu caste system? I mean, you don't just step off a boat and declare yourself a Brahmin priest.
You don't, which is exactly why their history is so opaque before the last millennium. They arrived as foreigners and settled in the coastal isolation of the Konkan region. They were entirely disconnected from the ancient Vedic traditions of the Desast.
Right.
However, historical texts, coupled with this genetic revelation, point to a fascinating sociological process, a delayed Brahminization event.
Brahminization being the process of social elevation and assimilation into the elite class.
Exactly. As this migrant community entrenched itself over a few centuries, they adapted rapidly. They became highly successful cultivators of cash crops. They were demonstrably literate and administratively skilled, and they integrated into the regional economy. Their skills became indispensable.
So they made themselves useful.
Very useful. Eventually, they were absorbed into the elite administrative layers of the local society. This culminated famously during the rise of the Maratha Empire. When Chipavans were brought into the royal courts, they served as administrators, diplomats, and were eventually elevated to the title of Peshwa, which effectively made them the prime ministers and de facto rulers of the entire empire.
It is a phenomenal story of adaptation. We have two distinct populations, the Disast and the Chipavan, sitting side by side at the absolute zenith of the social hierarchy. Yet they arrived there through entirely different millennia, utilizing completely different geographic vectors.
One walking across the Eurasian steppe, the other sailing across the Arabian Sea.
Exactly,
Yeah.
and carrying entirely different genetic blueprints. The caste system was supposedly a rigid, divinely ordained structure, but at the very top it proved dynamic enough to absorb a foreign population of Mediterranean sailors and elevate them to prime ministers.
It forces us to completely reevaluate our assumptions about how ancient social classes were formed and maintained. The elite tier was not a monolith created at the dawn of time and sealed in amber. It was a pragmatic, evolving structure. The Disast and the Chipavan share a social title, they share a rank, but their molecular logbooks are written in completely different genetic languages.
That perfectly covers the genetic paradox of the elites. But the Brahmins, for all their power, represent only a tiny fraction of the population. To understand the true biological engine of the subcontinent, we have to leave the administrative courts and the temples. We have to look at the vast, sprawling majority of the population.
The warriors and the peasants.
Right. And this is where the researchers apply the concepts of fission and fusion to explain the massive demographic movements of the masses. They analyzed two major groups to map this out. First, they looked at the Dangar.
Yes, the Dangar. In the traditional hierarchy, the Dangar are categorized as Shudras. They are a massive, historically pastoral and peasant caste. They were traditionally shepherds, blanket weavers, and agricultural laborers.
And the second group under the microscope.
The Maratha. They are the middle-ranking warrior and aristocratic class, the Kshatriyas we discussed earlier.
Okay.
But the Maratha are not just a small, elite military guard. They are demographic heavyweights. They are geographically dispersed across the entire region and currently represent over 50% of the total population in Maharashtra.
Wait, over 50% of the population belongs to the warrior caste?
Yes.
That alone suggests some incredible societal absorption. Let's start with the Dangar, though, the peasant class. When researchers sequenced their DNA, what did the foundation of the population look like?
The Dangar genome is a textbook study in the genetic process of fission. When the researchers looked at the deep ancestry markers, the Dangar displayed very high levels of ancient Proto-Asian genetic roots. Their Y chromosome data showed a tremendous amount of internal diversity, possessing a wide variety of ancient indigenous male lineages.
Okay, so deeply rooted locally.
Yes. However, their overall genomic profile was incredibly heterogeneous. They were not a uniform, isolated block of indigenous pastoralists.
If they are deeply rooted pastoralists who are theoretically restricted from marrying outside their caste, what was driving that genetic heterogeneity?
The researchers identified a striking pattern of asymmetrical gene flow. The Dangar retained those predominantly ancient South Asian male lineages, but their maternal lines revealed a surprising continuous influx of West Eurasian female lineages.
Wait. We established earlier that women were the genetic bridges due to patriarchality. But you're saying West Eurasian women, perhaps the descendants of incoming traders or migrants, were migrating downward into the lower-ranking indigenous peasant castes.
That's what the data shows.
That completely contradicts the idea that everyone was trying to marry up the social ladder.
It highlights the chaotic reality of human migration. Marriage patterns, especially on the fringes of urban centers or in rural agricultural zones, rarely adhere strictly to top-down theoretical rules. But the most defining characteristic of the Dangar is this ongoing process of fission.
The splitting apart.
Right. Over centuries, driven by changing ecological environments, periods of severe drought on the Deccan Plateau, or simply the biological necessity of finding new pastures for their massive flocks, this giant peasant cluster continually fractured.
Which makes sense. A pastoral lifestyle demands mobility and vast amounts of land. You can't keep a million shepherds in one valley.
Exactly. The researchers note that the original Dangar caste has splintered into at least 23 distinct endogamous subcastes. You have groups like the Hatkar, the Zende, the Ahirs, each specializing in a slightly different occupational niche or geographical zone.
It's like observing a massive river fracturing into a sprawling delta. A group of shepherds takes their flocks 50 miles away to a newly accessible plateau. Because travel is difficult, over a few centuries, they stop interacting and marrying with the original group. They become geographically isolated.
That's the mechanism.
And due to genetic drift, the random fluctuation of gene frequencies in small, isolated populations, that splinter group eventually becomes a totally separate, distinct genetic island, complete with a new subcaste name. The occupational necessity drives the genetic fission.
That is a perfect articulation of it. The Dangar represent a massive indigenous population constantly breaking apart into smaller, isolated fragments to survive the ecological pressures of Western India.
So if the peasants are defined by constant fragmentation and isolation, what about the Maratha, the massive warrior class?
If the Dangar represent fission, the Maratha represent the absolute pinnacle of genetic fusion. When the researchers analyzed the maternal DNA of the Marathas, they found a widespread distribution of Paleolithic mtDNA haplogroups. This is a critical data point because it means the Maratha warriors share the exact same deep, ancient Proto-Asian maternal ancestry as the Dangar peasants.
Ah, so their heirlooms trace back to the same ancient South Asian founding mothers.
Yes, that anchors them deeply to the indigenous population.
But the Marathas were the martial power of the region. They forged empires. What did the paternal lines, the surnames of the kings and warriors reveal?
This was perhaps the most fascinating paternal discovery in the paper. When analyzing the Y-DNA of the Maratha males, the researchers identified a remarkably high frequency of a specific lineage known as J2.
J2. I'm familiar with the J haplogroup family. Isn't J2 widely recognized as the primary genetic signature of the early agricultural expansions? Like, it's the lineage associated with the very first farming communities emerging from the Fertile Crescent and Anatolia.
That is correct.
How does the defining genetic marker of Mediterranean and Levantine farmers end up dominating the Y chromosomes of India's most formidable warrior caste?
It seems counterintuitive until you understand the historical mechanics of empire building in the Deccan region. The Marathas were never a single, pure royal bloodline descended from a specific ancient patriarch.
Okay.
The Maratha caste was a massive, pragmatic conglomeration. As various regional powers rose and fell, the descendants of ancient royal dynasties like the Mauryas and the Chalukyas, the rise of the Deccan sultanates, and eventually the sprawling empire of Shivaji, they needed manpower. You cannot maintain control over half the subcontinent without a massive army.
And to build a massive army, you can't be strictly exclusive about who gets to hold a spear.
Exactly. The Marathas aggressively assimilated incoming foreign warriors who arrived with the various invasions, but crucially, they also absorbed enormous numbers of the local agrarian classes into their martial ranks.
Oh, I see.
The farmers who carried that J2 agricultural lineage, which had slowly migrated into India from the Near East millennia prior, were recruited, drafted, or volunteered into the armies.
So the boundary defining the warrior caste was essentially entirely fluid based on utility. If a farming community picked up swords to defend their land, or if they were conscripted by a Maratha king and proved themselves in battle, they were no longer classified as farmers. They were socially elevated and biologically assimilated into the Maratha fold.
They are the ultimate genetic melting pot of Western India. They absorbed the agricultural communities carrying the Anatolian J2 lineage, they absorbed foreign mercenaries, and they blended all of them with the ancient indigenous maternal populations. The Maratha caste was less of a strict genetic lineage and more of a massive martial recruitment drive that spanned centuries.
This raises a massive structural question about the entire caste system. If the Dangar peasants are constantly fracturing and spinning off into new subgroups based on their jobs, and the Maratha warriors are constantly absorbing massive swaths of the farming population to fuel their armies, does the genetic data actually show them as separate, distinct classes? Or are the kings and the peasants just genetic cousins playing different social roles?
Answering that question is the ultimate objective of the entire study, and it requires us to zoom out entirely. We have to look past the individual, unbroken lineages of the Y chromosome and the mtDNA.
We have to open the complete recipe book.
Exactly. The recombining autosomal DNA. We have to look at the overall genomic synthesis of all 4 groups simultaneously.
Because up to this point, looking through the narrow keyholes of specific paternal and maternal lines, we've painted a picture of wild, disparate origins. The elite Deshath Brahmins are ancient Central Asians married to indigenous women. The Chitpavan Brahmins are Mediterranean sailors who adapted into administrators. The Dongar peasants are fracturing indigenous tribes absorbing Eurasian women. And the Maratha warriors are Anatolian farmers who learned to fight.
Right.
Based on those specific markers, it sounds like 4 completely unrelated species cohabitating in Maharashtra.
If you only looked at the non-recombining lineages, that is exactly the conclusion you would draw. They brilliantly trace the diverse, disparate geographic origins of these groups deep into antiquity. However, when Gaikwad and Kashyap analyzed the admixture, estimates the overall percentage of the different genetic ingredients that make up their complete modern DNA, a totally unified picture emerged.
Okay, I'm ready for the twist. What were the final admixture numbers across the board?
Across all 4 castes, from the highest-ranking Chitpavan Brahmin to the lowest-ranking Dongar Shepherd, every single group demonstrated a remarkably high shared indigenous genetic component.
How high?
Between 60 to 90% of their overall autosomal DNA is deeply rooted, shared South Asian ancestry.
60 to 90%.
Yes. The Western Eurasian admixture, the genetic echoes of the Central Asian charioteers, the Anatolian farmers, and the Mediterranean sailors only ranges from 10 to 40%, varying slightly depending on the specific group.
So despite the wild, multi-continental origin stories of their specific founding ancestors, the vast majority of their DNA today is shared, indigenous biology.
Yes, but the true, undeniable proof of this shared biology was found when the researchers ran an AMOVA.
An AMOVA?
AMOVA stands for Analysis of Molecular Variants. It is a statistical method used to determine where the most significant genetic differences actually exist. It asks a fundamental question. Is the genetic gap wider between the different castes, or is the genetic gap wider between individuals within the exact same caste?
Okay. Given the strict rules of endogamy and thousands of years of supposed isolation, you would assume the castes would be genetically distinct from one another. What did the AMOVA reveal?
The AMOVA revealed that the genetic variance within the populations was over 90%.
Wait, let me make sure I'm fully conceptualizing this.
Yeah.
The statistics dictate that there is a wider range of genetic difference between 2 random Maratha warriors standing next to each other than there is between a Satha warrior and the Brahmin priest officiating his ceremony.
That is exactly what the data demonstrates. The internal diversity of the groups completely eclipses the differences between the groups. But the researchers went one step further to quantify this definitively. They calculated the GST estimate for the biparental autosomal markers.
GST.
GST is the coefficient of gene differentiation. It specifically measures the level of genetic isolation between subpopulations. If groups are totally isolated from one another, say, humans on Earth and a colony of humans stranded on Mars for 10,000 years, with 0 interbreeding, the GST approaches 1, or 100%. If the populations are freely mixing and exchanging genetic material, the GST approaches 0.
Okay, this is the ultimate biological verdict on the caste system. What was the GST value for these 4 rigidly separated socially and religiously stratified castes?
The GST value for these 4 groups was an astonishingly low 1.1%.
1.1%.
1.1%.
Wow.
From a perspective of population genetics, a GST of 1.1% indicates a profound, almost total lack of genetic differentiation between these populations.
How is a number that low practically possible? We just outlined a social system that explicitly forbade intermarriage for centuries. You do not marry outside your caste. The social penalties for doing so were historically severe. How can they be 98.9% genetically intertwined?
Because the data proves that despite the strict, centuries-old ideological rules of social hierarchy, 2 forces consistently proved stronger: geographical proximity and fundamental human nature. A GST value of 1.1% is the biological fingerprint of substantial, continuous, and pervasive recent gene flow among all of these castes.
People were completely ignoring the rules.
Consistently and constantly. The paper highlights several mechanisms for this. They specifically point to substantial recent gene flow between the martial Marathas and the elite Brahmins through a process called hypergamy, or the forming of matrimonial alliances.
Ah. Political marriages.
Exactly. Remember the power dynamic. The Marathas were the kings, the generals, the physical power of the empire. The Brahmins were the elite administrators, the Peshwas, the intellectual power. Regardless of what the ancient religious texts dictated about caste purity, powerful families formed alliances through marriage to consolidate their influence. Power marries power.
And the autosomal DNA captured that political reality that documented the marriages that the official histories might have tried to obscure.
Precisely. Furthermore, the autosomal data revealed deep, shared recent ancestry flowing between the Maratha warriors and the Dongar peasants. It confirms that these were not isolated classes living in parallel universes. They were a biological continuum.
The continuum.
The peasant farmers elevated themselves into the warrior class during times of conflict. The warriors absorbed the farmers to build their armies, and the elites quietly intermarried to secure their political dynasties.
When you look at the totality of Gaikwad and Kashyap's research, what does this actually mean for us as observers of history? It seems to me that the paper proves a massive, incredibly beautiful paradox about human identity.
So,
Well, on one hand, if you look through the highly specific narrow lenses of the Y chromosome and mitochondrial DNA, the markers perfectly trace the wildly diverse, disparate origins of these groups. You can clearly see the arrival of the ancient Central Asian charioteers, the seafaring Mediterranean merchants landing on the Konkan coast, the Anatolian farmers pushing eastward, and the deep ancient roots of the indigenous pastoralists. The entire sweeping story of global human migration is accurately recorded in those specific lineages.
The specific lineages are the historical footnotes, but
But on the other hand, when you open the lens wide and look at the overall genomic picture, the 1.1% GST value, it shows that the reality of living next to each other, sharing the same monsoons, the same droughts, and the same river valleys in Western India, ultimately blended all of those disparate global travelers into one shared genetic family. The social architecture, with all of its rules, its walls, and its enforcement, completely failed to stop the biological blending.
it is a stunning, undeniable synthesis. The genetic toolkit brilliantly revealed the ancient Scytho-Dravidian roots of the Desas establishment. It uncovered the Irano-Scythian maritime arrival of the Chitpavan administrators, and it mapped the complex, ongoing fission fusion dances of the Maratha armies and the Dangar shepherds. But the Recombining Autosomal DNA, the complete recipe book, reveals the ultimate truth of human population genetics.
Geography consistently trumps ideology.
Exactly. If you live together in the same space for long enough, regardless of the rules you invent, you will inevitably blend together.
It really has been a phenomenal journey tracing this. We've gone from the freezing steppes of Central Asia to the ancient trade winds of the Konkan coast, entirely by looking at the microscopic chemical markers hidden inside ourselves.
As we conclude this analysis, there is a final, broader concept I want to offer for consideration. When we study the Indian caste system, we're looking at one of the most famously strict, complex, and socially regimented hierarchical systems ever devised in human history. It was meticulously designed, philosophically justified, and socially enforced specifically to keep different groups of people biologically apart.
Right.
Yet, as that 1.1% GST value mathematically proves, even a system that rigorous could not prevent the deep structural blending of human genomes over a few millennia, it failed at its primary biological directive.
It totally failed. The walls were illusions.
If a system that entrenched could not stop human genetic integration, what does that imply about the modern borders we obsess over today? We draw arbitrary lines on maps, we build physical walls of concrete and steel across deserts, we pass complex immigration laws, and we try so desperately to categorize, separate, and isolate ourselves based on nationality or ethnicity.
that's true.
But our DNA, the actual physical instruction manual of what it means to be human, suggests that human biology is fundamentally driven to connect, to mix, and ultimately to dissolve the very boundaries we try so hard to enforce.
That is an incredibly powerful thought to walk away with. History is not just a dry record of our divisions and our conflicts. Our bodies are the living, breathing proof of our inevitable connections. And it brings us right back to the premise we started with. A truest, most unedited, most accurate history book isn't sitting under glass in a museum. It is right there, pulsing through your veins. Thank you for joining us on this deep dive into the molecular past. Keep questioning the walls around you, stay insanely curious, and we will see you next time.